I would like to, first of all, thank Professor Natarajan and Dr. Kaushik for giving me this opportunity to present our work on glaucoma. So I'm going to switch gears. I'm not a clinician. I'm a PhD scientist working in a university. So I work with clinicians. And what I'm going to do is to show you some of our latest work in the next 10 minutes or so. How we have come up, it's a drug discovery kind of research, how we approach this from a pure biochemistry point of view. So I'm also thankful to Professor Natarajan for bringing me out of Denver, where I work, which is -2 or -4 in the midday temperature. Here it is balmy 22 or 24 degrees. I'm very thankful for that. So enjoying the warmth. So this work was done in collaboration with a group in University of North Texas Health Science Center, Dr. Dorota Stankowska and Dr. Raghu Krishnamurthy, who are experts on neuroprotection in glaucoma. So I don't need to show you this slide probably. For many of you are clinicians here. Glaucoma is, I'll quickly run through a few points. It's a heterogeneous group of progressive optic neuropathies. Approximately 64 million people worldwide have this disease. This will increase to 76 million in 2020 and 120 million by 2040. Nearly 2 to 3 million Americans above the age of 40 have the disease. The number is expected to increase by more than double from 2010 to 2050. There are many major risk factors. Some of them are listed here: age, myopia, family history of glaucoma. We heard a lot about glaucoma since morning, so I don't need to go through the risk factors again. The main consequences of glaucoma is slow and progressive death of retinal ganglion cells, which Dr. Harsh Kumar just mentioned, and optic nerve damage and visual field loss, which could lead to blindness. What are the contributing factors for RGC death and optic nerve damage? It's the disruption of axonal transport, astrocyte proliferation and hypertrophy, extracellular matrix remodeling, excessive nitric oxide production, elevated glutamate concentration, and neutrophil deprivation. So also recently several papers have come out to show that crystallins, which are the proteins abundant in the lens, they play a role. They are associated with glaucomatous optic neuropathy. I'll show you in a moment how we approach this. So one of the crystallins in the lens and in our body is AlphaB Crystallin. Okay, so this is a protein which is also present in retinal ganglion cells. As I said, this is a major protein in the lens and retina. It's a chaperone protein. That means it doesn't allow other proteins to undergo denaturation in cells. It's also an anti-apoptotic protein. That means it prevents cell death. If you stain human healthy retina and compare with the glaucomatous retina, what you see that this crystalline protein is highly expressed in the nerve fiber layer and the outer nuclear layer, but it's almost completely absent in a glaucomatous eye. Right? So this is the ganglion cell layer here. You can see some of it is lighting up for crystallines. So it's completely absent in glaucomatous eye. This means what? So when we saw this, we wanted to understand this protein in the context of glaucoma. So what we then did was used an animal model, which is AlphaB Crystallin. This protein is knocked out from this animal. If you look at the RGC number in these animals, this is central retina and the peripheral retina. This is the wild type. That means that has this protein, and this is the knockout that doesn't have the protein. In both central and peripheral retina, this is a retinal flat mount and stained with a stain called BRIN3A. This is a transcription factor expressed in retinal ganglion cells. So what you see, each green dot is a ganglion cell. So if you see the central retina in the wild type and the peripheral retina, there are lots of ganglion cells. But if you don't have this protein in the retina, you don't have as many ganglion cells. That means this crystalline protein is important for the survival of ganglion cells. Now we took a different an approach that we have been, this took us almost 6 years to develop. So there is a small segment of that crystalline protein that's enough to function, to have the two functions, that is the chaperone protein effect and also the anti-apoptotic effect of the protein. This is a segment of that protein from amino acid 73 to 92. So this we call, is a patented peptide now, we call this peptane 1. So we use this peptide, or now I'm going to refer this as peptane 1 from now on. Now you can culture retinal ganglion cells, rat retinal ganglion cells, have them in culture and expose to hypoxia to induce death. Okay, so when you have, when you expose these cells to hypoxia, they die. This is the dead, the red dots are dead cells here. Now if you have our peptide, then you don't have the red dots at all. So we are just adding our peptide to cells that are growing in cell culture, and when those cells are exposed to hypoxia, these ganglion cells are protected by our peptide. Now, we also then asked, this is all cell culture, so what, right? So we took a rat, we can culture rat retinal, whole retina, that we call that retinal explants. Again, we can have the explants in normoxia or hypoxia. When you expose the explants to hypoxia, retina cells, the ganglion cells die. Again, we are staining the ganglion cells with BRIN3A. So the green dots are ganglion cells here. So normoxia, you can see there are lots of cells. This is a scrambled peptide. That means a control where it doesn't have the same effect that our peptide has. So that in the presence of that peptide, cells are dying. But if you have our peptide, peptane 1, not all cells are dying. You can see cells that are dying here are protected by our peptide. So our peptide is protecting in a ganglion cell culture and also in the retinal explants. So this led us to this study when Dr. Morrison developed this animal model for glaucoma. You can inject a hypertonic saline solution into the episcleral vein of the eye, and the intraocular pressure in these animals can raise to about 30 millimeter mercury, and you can keep these animals for a month or so. And then what we did was we injected our peptide 3 times a week at 100 micrograms per injection intraperitoneally. Mind you, this is not intravitreous. So this is intraperitoneally to test the concept whether this peptide can protect ganglion cells in the retina. So what we then did was to retrograde labeling of the ganglion cells by injecting fluorogold into the superior colliculus, and then you can, after 3 months of the injection, you can sacrifice the animals and look at their retina. So the red dots here are the ganglion cells in the retina. So if you inject a scrambled peptide that is not functionally active, you can see when your IOP is elevated, the ganglion cells are dying. That is this death here. But if you have our peptide injected intraperitoneally into these animals, then you can protect the ganglion cells. So so we tested not only in cell culture and retinal explants, but also in one model of glaucoma. Now, what's even more striking, when I saw this, I was really shocked that this peptide, when we inject, in the same animals, if you look at the axons of the ganglion cells, we can label retinal ganglion axons with this paraphenyl diamine. So I don't know whether you can see from there these white dots here. By canaliculating the anterior chamber with PBS, which is introduced to the anterior chamber through a reservoir which is kept at an elevation. So what happens then is the intraocular pressure raises to 70 millimeter mercury, and we keep that pressure for 60 minutes and take out the needle. So this causes massive reperfusion injury. So again, if you see central retina and the peripheral retina, this is central retina and the peripheral retina. If you inject the scrambled peptide, then the cells are dying here. So the peptide is ineffective. But if you inject our peptide into these animals, again intraperitoneally, this is not injected intravitreally. Intraperitoneally, it's protecting. Both, you can see here, the peptide is protecting the ganglion cell in this acute model. This is an acute model. We don't have to wait too long. 14 days after IR injury, we can see massive amounts of ganglion cells being gone from the retina. But our peptide is working even in this acute condition. If you look at the apoptosis of ganglion cells in the retina in these animals, after 14 days of this ischemic injury, this is a contralateral eye as the control. There is no dead cells. These are the green dots these are ganglion cells. They are dying by apoptosis. If you inject our peptide and stress these animals to IR stress, then they don't have no apoptosis at all, zero apoptosis. This was really, really surprising to us. A peptide injected intraperitoneally is protecting the ganglion cells in an acute model of ischemia. If you do the ERG on these animals, sorry, I skipped. ERG on these animals, you can see this scotopic ERG, A and B wave, both. This is reduced. This is the A wave. The red line is the ischemic injury to the retina reduced, and this is the B wave reduced. But if you inject our peptide to these animals intraperitoneally, this curve shifts upwards. That means the retina is protected. Both the photoreceptor cells and bipolar cells are protected. The one last experiment that we did, this is even more surprising to us. This is the axonal transport. So in the basic science world, the axonal transport has become a real big thing in glaucoma research, with how to protect the axons of the ganglion cells. So again, we have the scrambled peptide and the real peptide. We are now injecting a chlorotoxin subunit B into the vitreous of the ischemic retina to see how far the chlorotoxin travels along the axis of the ganglion cells. So this is done after 2 days after injection of the chlorotoxin to the eye. So you can see it's obviously labeled with the fluorescence dye. If you have the scrambled peptide, this is from the eye to the brain here. This is not traveling much. But if you have our peptide injected into the eye, then the axon is protected. You can see this dye is traveling well along the axon. So again, telling us our peptide is very effective against ganglion cell, both the soma and the axon transport defects that you see. It can protect both of those. Now, I told you we are injecting the drug into the intraperitoneal cavity. How do we know that it goes to the retina, right? A lot of people question this. It's a small peptide. It should be taken up by the liver and metabolized within minutes. But it turns out this one goes to the retina. If you do a flat mount, this is the serum concentration of the peptide in the retina. You can see within 3 hours it comes down, peaks at about 1 hour. But if you take out the retina and do a flat mount, you can see the retina is fully, it's in the retina, it's distributed throughout. If you do a cross section of the retina, you can see our peptide gets distributed across the retina and somewhat some in the ganglion cells as well. So we know that the intraperitoneally injected peptide crosses the blood-retinal barrier, goes in the retina and protects ganglion cells. One last slide. So we are now working with a group of nanotechnology people to put this peptide in a nanoparticle solution and see whether we can subconj injection, we can make it release on a sustainable manner, so we can prevent ganglion cells, at least in the acute models and also in the more recent model of glaucoma. So in summary and conclusion, peptide 1 crosses the blood-retinal barrier. This is the first for a peptide to have this property. It protects ganglion cells, axons in animal models of glaucoma. It improves the ERG response in the animal models of glaucoma. So peptide 1 could be a novel therapy against neuronal death in glaucoma. I find probably the best use for this peptide is probably the acute angle closure glaucoma and in traumatic eye and head injury. This could be very useful in these situations. So I just want to acknowledge the people who worked with me. These are the postdocs who worked with me for many years. Dr. Shruti Sampath Kumar, she did her MD here, and she came to my lab to work on this project. Dr. Meena Pancheva is our clinician. She helps me with clinical evaluations of data. And this work was supported by Research to Prevent Blindness, New York. So with that I stop, and if you have any questions, you can answer. Thank you.